Atmic Biotech Solutions Private Limited.
Virus COVID-19, it is a RNA virus. This is the organization of the virus. Genetic material is RNA, which is surrounded by a protein envelope. The protein envelope has various proteins like membrane protein, envelope protein, spike protein, nucleocapsid protein. And in RT-PCR, for the detection of COVID-19 virus, the nucleocapsid gene N is used for the detection of COVID-19 viruses. One of my previous video I have discussed about normal PCR. The only difference between normal PCR and real-time quantitative PCR is here we are using a double-stranded DNA binding type. For example, cybergreen. This will bind with the double-stranded DNA and emits fluorescence only when it is bound to a double-stranded DNA. So thus we can monitor the product formed in real time. That is the advantage of real-time quantitative PCR over conventional PCR. These are the steps for the detection of COVID-19 viruses. First one is sample collection, second one is RNA isolation, then RNA has to be converted to DNA using cDNA synthesis, then RT-qPCR, then data analysis. So the samples collected from the nasal or throat swab, the RNA will be isolated using phenol and guanidinium isothionase, then RNA extraction is done. Next step is reverse transcription. We have to convert this RNA into cDNA. Then the cDNA will be used as a template for RT-qPCR using nucleocapsid N gene specific primers. And the data will be analyzed. So this is the output of qPCR and this is known as qPCR curve. Here you can see a CT value or cycle threshold. It is defined as the number of cycles required for the fluorescent signal to cross the threshold. In the reaction mixture, we are providing the fluorescent dye, so there will be a background fluorescence. Only when our target gene is present in the reaction mixture, then only CT value will form, or during reaction after the number of cycles, a certain number of cycles, the fluorescence will go beyond the CT value, that is threshold value. So if a sample is positive, a sample is strongly positive for that particular gene, the CT value will be less than or equal to 29. If a sample got a CT value less than or equal to 29, we can say that that patient is strongly positive with COVID-19 infection. If it is between 30 and 37, it is moderately positive, then 38 and 40, we can say that the patient is weakly positive. And if there is no CT value, that means that patient is negative for COVID-19 infection.